In today's lecture, we are going to discuss the synapse. Definition: synapse is the junction between two neurons. It is not an anatomical continuation, but a physiological continuity between two nerve cells. Classification of synapse. Synapse is classified by two methods: anatomical classification and functional classification. Let's discuss the anatomical classification of synapse first. Depending upon ending of axon, synapse is classified into three types: axoaxonic synapse, in which axon of one neuron terminates on axon of another neuron; axodendritic synapse, in which the axon of one neuron terminates on dendrite of another neuron; axosomatic synapse, in which axon of one neuron ends on soma or cell body of another neuron. Functional classification. Functional classification of synapse is on the basis of mode of impulse transmission. According to this, synapse is classified into two categories: electrical synapse and chemical synapse. However, generally the word synapse refers to a chemical synapse. Electrical synapse. It is the synapse in which the physiological continuity between the presynaptic and the postsynaptic neurons is provided by gap junction between the two neurons. There is direct exchange of ions between the two neurons through the gap junction, due to which synaptic delay is less. This type of impulse transmission occurs in some tissues like the cardiac muscle fibers, smooth muscle fibers of intestine, and the epithelial cells of lens in the eye. Chemical synapse. It is the junction between a nerve fiber and a muscle fiber, or between two nerve fibers, through which the signals are transmitted by the release of chemical transmitter. In the chemical synapse, there is no continuity between the two neurons because of the presence of a space called synaptic cleft between the two neurons. Functional anatomy of chemical synapse. Neuron from which the axon arises is called the presynaptic neuron, and the neuron on which the axon ends is called postsynaptic neuron. Axon of the presynaptic neuron divides into many small branches before forming the synapse, called axon terminals. Axon terminals are of two types. First, terminal knobs, which are enlarged and are concerned with excitatory functions of the synapse. And second is the terminal coils or free endings. These are wavy or coiled with free ending without the knob. These terminals are concerned with inhibitory function. Presynaptic axon terminal has a definite intact membrane known as presynaptic membrane. Axon terminal has two important structures: mitochondria, which help in the synthesis of neurotransmitter substance, and synaptic vesicles, which store neurotransmitter substance. Membrane of the postsynaptic neuron is called postsynaptic membrane. It contains some receptor proteins. Small space in between the presynaptic membrane and the postsynaptic membrane is called synaptic cleft. The basal lamina of this cleft contains cholinesterase, which destroys acetylcholine. Functions of synapse. On the basis of functions, synapses are divided into two types: excitatory synapses, which transmit the impulses, excitatory function, and inhibitory synapses, which inhibit the transmission of impulses, inhibitory function, excitatory synapse. When an action potential reaches the presynaptic axon terminal, it triggers the opening of voltage-gated calcium channels in the presynaptic membrane. Calcium ions from the extracellular fluid enter the axon terminal. This influx of calcium causes the release of neurotransmitter molecules from vesicles by exocytosis. An excitatory neurotransmitter, such as acetylcholine, is released into the synaptic cleft and crosses to the postsynaptic membrane. There, it binds to receptor proteins on the postsynaptic membrane, forming a neurotransmitter receptor complex. This complex opens ligand-gated sodium channels, allowing sodium ions from the extracellular fluid to flow into the postsynaptic neuron. Since sodium ions carry a positive charge, the resting membrane potential inside the postsynaptic neuron becomes less negative, leading to a small depolarization. This small depolarization is called an excitatory postsynaptic potential. Excitatory postsynaptic potentials are local, non-propagated membrane potentials that occur at the synapse. If the excitatory postsynaptic potential is strong enough, it will open voltage-gated sodium channels in the initial segment of the axon. Sodium ions then rush in, causing further depolarization, which triggers an action potential. The action potential then travels along the axon to other segments, propagating the signal. Inhibitory function. Inhibition of synaptic transmission is classified into 5 types: postsynaptic or direct inhibition, presynaptic or indirect inhibition, negative feedback or Renshaw cell inhibition, feed-forward inhibition, reciprocal inhibition. But here we will discuss postsynaptic and presynaptic inhibition only. Postsynaptic or direct inhibition. When the action potential reaches the axon terminal, it causes voltage-gated calcium channels in the presynaptic membrane to open. Calcium ions enter the presynaptic terminal from the extracellular fluid, which triggers the release of neurotransmitter from synaptic vesicles through exocytosis. The inhibitory neurotransmitter, commonly gamma-aminobutyric acid, crosses the synaptic cleft and binds to GABAA receptors on the postsynaptic membrane. This binding opens ligand-gated chloride channels, causing chloride ions to flow into the postsynaptic neuron. In some cases, GABA can also activate GABA-B receptors, leading to the efflux of potassium. Both processes result in hyperpolarization of the postsynaptic membrane, generating an inhibitory postsynaptic potential. This hyperpolarization leads to the inhibition of synaptic transmission. Presynaptic or indirect inhibition. Presynaptic inhibition occurs due to the failure of presynaptic axon terminal to release sufficient quantity of excitatory neurotransmitter substance. Presynaptic inhibition is mediated by axoaxonic synapse. In spinal cord, a modulatory neuron called presynaptic inhibitory neuron forms an axoaxonic synapse with the presynaptic neuron. This inhibitory neuron inhibits the presynaptic neuron and decreases the magnitude of action potential in presynaptic neuron. The smaller action potential reduces calcium influx. This in turn decreases the quantity of neurotransmitter released by presynaptic neuron, so the magnitude of EPSP in postsynaptic neuron is decreased, resulting in synaptic inhibition. That concludes our lecture for today. See you all next time.